Welcome. You're here for a deep dive, aiming to understand complex topics efficiently. Today, we're looking at new research based on a study and a related news report from April 13th, 2025. The focus is on colorectal cancer and a critical question. Why does it sometimes spread to the brain? This is often a devastating turn. Our mission is to unpack the key genetic findings from this research and consider what they might mean moving forward.
Let's start with the context. Colorectal cancer has a major global impact. We're looking at roughly 900,000 deaths worldwide each year because of it. When the cancer metastasizes, meaning it spreads, the situation becomes far more serious. About a quarter of patients already have metastases when they're first diagnosed, and another 20% or so will develop them later on.
And the common sites for this spread are usually the liver and lungs.
That's correct. Those are the most frequent locations. Brain metastasis is rarer, but it brings unique and severe challenges. Understanding the reasons behind it has been a long-standing research goal.
This brings us to the new study from the University of Augsburg. Researchers there analyzed quite a number of tumor samples, over 300, and importantly, this included 39 samples specifically from brain metastases that allowed for direct comparison. What was the central discovery from this genetic analysis?
The main finding is quite striking. Tumors that spread to the brain carry a significantly higher number of genetic alterations. This is compared to colorectal cancers that spread elsewhere, or even the primary tumors. This accumulation of genetic changes seems characteristic of cancer cells able to thrive in the brain.
And one particular gene, KRAS, seems central to this. KRAS is already known to be a driver in many cancers, affecting cell division speed. What was the specific finding about KRAS in the context of brain metastases?
In the brain metastases studied, KRAS often showed two things happening together. Not just alterations, which are somewhat common in colorectal cancer, but also multiplication. This is technically called amplification, multiple copies of the altered gene. Think of it as turning up the volume on a faulty signal, telling cells to grow uncontrollably.
So it's the combination, the alteration, and the amplification that's particularly potent.
It appears so. This combination seems to fuel a much more aggressive growth pattern. The study lead, Professor Monica Golas, specifically highlighted changes in KRAS linked to this increased aggressiveness.
Does this aggressiveness also tie into how these tumor cells produce energy?
Yes, the study found a related metabolic shift. These aggressive tumors tend to rely more heavily on sugar, on glucose, for energy, rather than using oxygen efficiently. This shift towards glycolysis is actually a known characteristic of many aggressive cancers.
So this research starts to give us clues about the why the brain question. It suggests adaptation.
It offers the first steps towards an answer. The findings indicate that cancer cells can adapt genetically to specific organ environments. The distinct genetic patterns in brain metastases suggest a sort of predisposition for spreading there, rather than the liver or lungs. It's what researchers term organ-related metastasis.
The timing of when these genetic changes occur seems important too.
It does. For brain metastases, these crucial genetic changes often seem to appear relatively late in the disease progression. Sometimes this can be years after the primary colorectal cancer was first diagnosed. This points towards an evolutionary process within the tumor population over time.
Was KRAS the only gene showing this amplification pattern in brain metastasizing tumors?
No, it wasn't the only one. The researchers also observed frequent multiplication of other genes involved in cell growth control. Genes like MDM2 and CDK4 were also often amplified in these specific tumors. These genes are known to accelerate cell division and spread, adding to the aggressive nature.
And just to reiterate, the overall number of genetic changes was markedly higher in brain metastasis.
Yes, significantly higher, not just compared to the original primary tumor, but also when compared directly to metastases found in the liver or lungs. This high number of alterations likely makes them harder to treat. But on the flip side, it might also make them more identifiable using advanced genetic tests.
The study methodology was key here, using advanced techniques.
Absolutely. They used modern analysis methods capable of detecting subtle genetic variations. And a critical point. The samples came from patients who hadn't yet received targeted therapies. This provided a clearer picture of the tumor's natural genetic evolution towards metastasis without treatment interference.
Understanding these genetic details surely opens up possibilities for treatment.
That's the hope and the direction this points towards. By identifying these specific genetic vulnerabilities in the cells prone to brain spread, the research could pave the way for earlier risk assessment. Identifying patients at higher risk soon could lead to developing more targeted therapies. treatments designed specifically to counteract these weaknesses.
And the scale of the analysis adds weight. Over 3,800 cases were looked at overall.
Correct. While brain metastasis itself is less common, analyzing this large cohort confirms its distinct and complex genetic landscape when it does occur. The crucial takeaway is that these specific genetic signatures associated with brain metastasis are measurable. That measurability is key for potential clinical application.
Professor Golas herself underscored this significance.
Yes, she stated that their work deepens the understanding of the biological mechanisms behind metastasis. And importantly, it pinpoints actual vulnerabilities in these tumor cells. This brings the possibility of earlier detection and tailored treatments much closer.
So let's recap the main points for everyone. Colorectal cancer spreading to the brain tends to happen later in the disease course. These metastases are marked by a high number of genetic changes, particularly involving the KRAS gene, which is often both altered and amplified. These changes drive aggressive growth and a shift in metabolism favoring sugar, which seems suited for the brain environment. The good news is that modern methods can detect these patterns, potentially early on. This opens the door for more precise therapies targeting this specific, challenging form of metastasis. It gives a much clearer picture than just general information about cancer spread.
This study provides truly vital insights into a complex biological process, highlighting specific genetic targets.
It certainly does, and it leaves us with something to consider. If distinct genetic changes drive metastasis to specific organs like the brain, what does this imply more broadly? How might this change our approach to cancer treatment and personalized medicine overall? Should we be thinking beyond just the primary tumor's genetics to anticipate and target the genetic drivers of spread to particular secondary sites?
Right.
Definitely some significant implications for future cancer care raised by this research.